Okay, so in this video we're going to look at how, basically protein synthesis. Now protein synthesis is how we're getting from the code of how to make proteins in our DNA to an actual protein. And it has two parts. It has transcription and translation. And so let's go ahead and see how this happens. So in general, we have our DNA our DNA has genes, and our genes are basically the protein coding regions. So a gene codes for a protein. Now before a gene, we have what's called a promoter sequence, and that's where we find the TATA box. So genes are protein coding regions of our DNA. So when we look at this, genes are the directions for making a protein, and how we make a protein has two steps. So there's transcription, which is copying DNA into RNA, and then there's translation, which is reading that RNA into a protein. So in the first step of transcription, where we're going to copy the code from DNA into RNA. So the enzyme that's used is RNA polymerase. Now RNA is single-stranded. It has the nucleotides A, U, C, and G. There is no T in RNA. And so basically you have this template strand of DNA that will act as a guide for RNA polymerase to build the complementary strand of RNA. So the side that is used to make the RNA is called the template strand. And this RNA that's made is the primary transcript, or the first RNA. So let's go ahead and see a little bit more about this. So as the primary transcript is made, if it's moving in this direction, RNA polymerase can add to that 3' end. We have what's called the template strand, is a strand of DNA that's being used as being transcribed and being used to build the code of RNA. You'll notice that there's a T in DNA, but when you think there should be a T in RNA, it's a U. The strand that's not being transcribed is called the coding strand. So now here we have the primary transcript, which is basically the pre-mRNA before it has been edited. So when we look at how this starts though, let's go ahead and look at eukaryotes. So in order to start transcription, eukaryotes are going to regulate when they express these genes. So in our DNA, we have promoter regions. That is where RNA polymerase attaches to begin transcription, because here's our protein coding region. Now way far upstream, we have an area called the enhancer region. The enhancer region is where very specific activators attach. So while every single cell in your body has the exact same DNA, so they'd have the same promoter sequences, genes, enhancers in every cell. But what's unique are the activators. So if this was a gene to make the enzyme salivary amylase that you use in your saliva or your spit, these would be the activators to turn that gene on in my salivary glands. While I'd have the same enhancer region, promoter region, and protein coding region, I'd have the same ones all in let's pretend my eyeball cells. I don't have these activators there. So yes, I'll have all of these sections, but if I'm missing the activators, then this gene will not be expressed in my eyeball. So really it comes down to the transcription factors in eukaryotes. So when specific activators attach in eukaryotes, it causes the DNA to bend, and it brings the enhancer region closer to the promoter region. Here's the gene, the protein coding area. And now we have some general transcription factors that will kind of help hold everything together, so RNA polymerase can attach to the promoter sequence. So this right here is the promoter. And then this is our gene. And then RNA polymerase can begin transcription. And then RNA polymerase will start to move down the DNA and build that complementary sequence. Now, if we are looking at eukaryotes, after we make our primary transcript, there is going to be some RNA processing. So RNA processing will occur, and in RNA processing, our pre-mRNA, our first mRNA that's made before it's edited, we have introns and we have exons. So exons are the parts that we're interested in, and that's the part that will be expressed. The intron are the parts that are spliced out. So as we go through RNA processing, there's 3 steps involved. We're going to splice out the introns, add a 5' cap, which is a modified guanine, and a poly A tail. So this poly A tail will kind of, as this RNA enters the cytoplasm, this will be degraded and hydrolytic enzymes will break it down. Now this mRNA is going to leave through a nuclear pore and leave the nucleus into the cytoplasm. So when we go into the cytoplasm though, let's go ahead and see what happens. Now it really depends. There's 2 fates here. So again, we have our nucleus and our cytoplasm. Now this represents the referer, and this out here is a cytoplasm. And the ribosomes I've highlighted in blue for us. So we have 2 kinds of ribosomes. You have bound ribosomes and you have free-floating ribosomes in the cytoplasm. If the RNA leaves the nuclear pore and attaches to a ribosome that's free-floating, it'll make a protein that will stay within the cytoplasm. However, if the RNA is translated on a bound ribosome, the protein that's produced at the referer is going to then travel in a vesicle to the Golgi, where it's going to be packaged, modified, sorted, like basically its final shape will be determined. And then it'll get carried in a vesicle to the cell membrane. Now at this point, there's 2 possible fates. One, it can be sent out by exocytosis. This may be a protein like insulin that will then travel in the blood to its target cells, or it may become possibly a receptor protein, or like a ligand-gated ion channel, could be a tyrosine kinase, could be a G protein, could be a channel for facilitated diffusion. But the point is here, it becomes part of the membrane, and that is still going to be part of this endomembrane system. So this here is basically the pathway that of protein synthesis for proteins that will be sent out of the cell by exocytosis or become membrane proteins. So when we look at the 2 fates of proteins, they either are made by free ribosomes staying within the cell, or they're made by ribosomes located on the referer. So this mature mRNA is made in the nucleus, but it doesn't go much further than the ribosomes. Once it's been used and translated, it's going to be degraded. There's enzymes that will break down the messenger RNA, and it's the final protein product that matters. So let's go ahead though and zoom in and see what's happening at this point. When the messenger RNA is on a ribosome, what does that even look like? So here we have, let's go back. Okay, so here we have some messenger RNA, and this is where the genetic code is used. When I mention the genetic code, this is what I'm talking about. So here, this piece of RNA in a eukaryote, in us, has already been edited. This is a combination of exons. And when we read RNA, we read it in groups of 3 called codons. So every 3 nucleotides represents 1 codon, and that's how you read this chart. So AUG for methionine is our first codon. So we have these things called tRNAs that stand for transfer RNAs, and their job is to transfer and bring the appropriate amino acid. So while my first codon is AUG, that codes for methionine, this purple amino acid would be met, or methionine. My next codon is CAU, so then I go CAU, U, it would be histidine, etc. Now, the ribosome is going to actually move down this messenger RNA. So AUG has already been used as a codon. Now this orange one that had histidine is here, and now this next codon, CCA, is where the next tRNA has arrived. Now I would use a genetic code, and CCA is going to be proline. So then proline would be brought. And now the tRNAs are free floating. Now this tRNA up here that coded for MET, after it's used, it is missing the amino acid, and that's why you eat protein, to replace these amino acids. And you can also make your own amino acids as well. Anyway, the ribosome will continue to move down, codon by codon, building this polypeptide tail. So as it moves down, it builds peptide bonds. So this is a peptide bond built between them. And eventually it's going to get to a stop codon. That stop codon signals the end of this protein. I only have 5 here to form a polypeptide, but in reality, an average protein in us would be about 400 amino acids. Now, this is also where mutations matter, because if a mutation were to change this like codon, for example, like here we have UUU codes for phenylalanine. Well, if a mutation had changed this to UUA, well, now that would be leucine. So this is where mutations have an impact on the structure of this polypeptide. So in my other drawings, I drew proteins like this, but really it's a chain of amino acids, which is the primary structure of a protein. Now, the cool thing in prokaryotes is that they don't do RNA processing. Oh, by the way, this is translation. The ribosome is translating the RNA into a protein. Okay, so in prokaryotes, they have their one circular chromosome. They have ribosomes. Their RNA polymerase will build the messenger RNA, which is green. They do not do RNA processing, so as soon as the mRNA is being produced, ribosomes can begin to build their proteins. The transcription, which is this part, and translation can happen simultaneously in prokaryotes. Then, to kind of summarize everything for us, what we see here, DNA has the code for life. However, that code for life is in the form of proteins. So to take that code, we do 2 steps. Protein synthesis is basically transcription and translation. So transcription is taking the DNA, in us, it would be in the nucleus, and transcribing or copying the DNA into RNA. And then we go through RNA processing, where we cut out our exons. Oh my gosh. We cut out our introns, splice together our exons. We use activators and general transcription factors to all make transcription happen. We get our messenger RNA that during the process of translation on a ribosome, the ribosome will read this mRNA in groups of 3 called codons. Now, these ribosomes can either be free if they're going to make proteins that stay within the cell, or they'll be bound to the ER if they're going to be part of the endomembrane system and end up at the cell membrane, either to become a membrane protein or to be sent out of the cell. Translation occurs in the cytoplasm, not the nucleus, relies on tRNAs to bring amino acids, and uses the genetic code in order to know which amino acid to bring. That is a summary of protein synthesis.